Thank you, everyone, and thank you for inviting me. It's a pleasure to be here. You couldn't ask for a more beautiful place. I'm going to hop right in. I have no conflicts of interest to disclose. I want to acknowledge the collaborators, postdocs, doctoral students who make this work possible, as well as the sponsors. The one that I'm going to be referring to in today's talk is right here. This is DoD-based work, and I'm going to jump to the end first. The DoD is a very interesting funding agency. They, as much as anyone else, are interested in addressing discontinuities and in making huge leaps in scientific discovery. But they have become increasingly risk averse, as many other funding agencies have, and they dictate translational efficacy in humans. And so much of our work has basic scientific components, but it also has translational components, and that's what I'm going to talk about today. Obviously, others have been successfully funded. This isn't authoritative, but we've had some success, and so I want to talk about what we're up to and what we're thinking about right now. Our study designs are modular. There are core questions, but there are then distinct sub-questions and sub-studies that take place at different sites with different groups of participants, various applications, and the study designs that I'm going to overview today, they're observational, they're experimental, there are basic applied and translational components, and partnerships. We visited the Wright-Patt Air Force Base a few months ago, and they were amazed that we were able to study one of the populations that I'm going to discuss. And they asked, well, how are you able to do this? And I said, well, how are you not able to do this? You're the military. But it's amazing. There are fiefdoms, there are silos. These things just develop over time organically. And my answer was it took a long, protracted effort with us visiting again and again and again, visiting with the commanders, visiting with directors, medical personnel, then becoming comfortable with us, knowing that we were there to help and that we wouldn't impede their normal operations. And so I'll talk about some of that. And obviously there are a lot of common themes here. So we'll get to what Dr. Jones was discussing with some of our TMS work, what Dr. Gilbert was discussing with the F10K clearance process. I mean, the work that we're doing is related to physiological resilience. One of the key things that we need to do is define it. Whenever you exercise, you're performing the world's oldest form of regenerative medicine, right? It's self-regeneration. People go through basic Army combat training. They go through the Navy SEAL selection screening course as midshipmen. Some of them pass, some of them don't. They're doing similar activities. So the question is, why are some of them suffering stress fractures, tendinopathy, chronic musculoskeletal strains and sprains? Others adapt just fine. They do well, right? How do they differ? I mean, resilience is a common term in the cognitive neuroscience and psychology fields, but it would apply to any other tissue as well, right? Why not? Every tissue, just about, adapts to stress. Some adapt well, some adapt in a maladaptive fashion, and the ones that adapt maladaptively, well, that winds up being an injury, and it's hugely costly to the military, and so it's something that we're focusing on. I mean, we're defining it based on whether or not people complete the training, whether or not people drop on request, the diagnosis of a stress fracture or not, or the incidence of a behavioral health event. There are a lot of different ways to look at it. I mean, just some examples, whether an individual has a specific genetic matrix metalloproteinase or a specific collagen isoform. These things are predictive of musculoskeletal injury. We know that chronic psychosocial stress predicts musculoskeletal injury. We're not sure if they produce more errors, if their movements are inaccurate, if the systemic milieu from a neuroendocrine standpoint prevents tissues from healing as they should. And we can also look at it from a systemic basis, whether there's an inflammatory immunophenotype, protective immunophenotype, the right place, the right time, as well as the mechanical properties, epigenetic modifications, and trophic factors that are secreted by muscle and other tissues in response to physical stress. So in the first module, which is how I would posit these, we are looking at basic Army combat training. It's about 10 weeks long, and it's physically brutal, physically exhausting, and we want to be able to predict stress fractures, strains, sprains, all these other musculoskeletal events. And this statement really encapsulates the notion well. I mean, individual biomarkers haven't fared all of that well. So we're thinking that there is a physiological signature, a profile, which is made by a combination of numerous biomarkers, and that might be indicative of a healthy state, an unhealthy state, progression towards recovery during rehabilitation, or a continued pathological process despite the resumption of strength, range of motion, all of the clinical measurements that we typically get. And this kind of technique and thinking has led to some promising results. Back to the key point slide. We're collaborating with Nathan Yates, who invented the differential mass spectrometry technique. He's at the University of Pittsburgh. And the benefit here, which was something the military was excited about in this otherwise really basic scientific platform, is that it doesn't require sample pooling. It doesn't require mixing. We can take individual samples from participants and we can do so repeatedly over time. That has huge value to the military from a clinical perspective. It's high resolution, it spans 5-6 orders of magnitude, and it's relatively fast. And from a clinical standpoint, reproducibility and rigor are emphasized here, lots of technical and biological replicates. We'll also collaborate with mathematicians to use machine learning to decode what we're detecting in the proteome to categorize individuals who are resilient and who are not resilient. And I'll get to that in just a second. Right now, we're at 400 proteins roughly. This is in the urinary proteome. We could get into detail on that if you're interested, but the goal is to hit about 1,500. And so we have some work to do, but the collaborative approach convinced the military that we would be able to pull this off. And really what that means is the optimization of collection and preservation techniques, better concentration and purification techniques, as well as quantification. And of course, the mathematics and statistical approaches that come on the back end. And so you can see the objectives here. Obviously, there is a big clinical impact if we can make this work in this population. We can discover new biomolecular profiles that predict or discriminate resilience. There are a number of collaborators who are interested in the effects of contraception, the effects of vitamin D synthesis, the effects of tobacco use, all of these other issues that serve as comorbidities and contributors to musculoskeletal injury. And so that was another way to enhance buy-in. And then from a basic standpoint, which the military may or may not care about, but we do, we'll have the opportunity to probe the size, chemical modifications, and relationships of the urinary proteome. We'll be able to expand that literature. So you can see there's a translational component, there's a basic scientific component. We can do it all in one in humans. So this is the general experimental approach. 4,000 people will come in, we'll oversample women, they'll be equally represented in relation to men. We'll take urinary samples before basic Army combat training. I just picked week 3 because that's when injuries tend to really present themselves. But we'll take urine samples throughout, and then at the end, and we'll surveil their medical records for 2 years after the completion of basic Army combat training to look at outcomes, right? And what will wind up happening is that we'll accumulate participants. It's an N of 4,000, there's a lot, right? At the end of the pipeline, some of them will be identified as resilient, some of them will be identified as non-resilient, based on a combination of factors, the factors that are of highest interest, the potential independent factors. That's a collaboration that we need to have with the military. We haven't even determined how we're going to do that yet, but we know that one way or another they'll be characterized. Then we'll run them through the proteomics pipeline. We'll use the machine learning, the mathematical approaches. We'll characterize participants. We'll rerun the study in a separate cohort. And based on what we found, we'll see whether our analytics can predict resilience or non-resilience, right? And that's what we're doing for that study. So for the second module, we're looking at acute resilience. The Navy SEAL screener program is really interesting because it has high rates of attrition and injury. And yet, if individuals, these are midshipmen at the Navy Academy, if they pass this program, they are overwhelmingly more likely to wind up becoming Navy SEALs compared to the other points of entry. So there's something magical about this program from a predictive standpoint. And obviously, it can be improved, but it's getting at the right underlying features. Okay, we don't really understand the physiological and physical cognitive impact of the screener, so we're going to do that, and we also want to refine the algorithm to see if psychophysical stress, in this acute scenario, can be represented or in any way tied to what we're detecting on a chronic basis, and we have no idea at this point. But we're talking about similar measurements. we can validate our algorithm, we can look at specific traits, and we can determine the nature and extent of the stress. This will be the experimental protocol. Once again, individuals who fail or drop on request will be distinguished from those who pass. We'll acquire samples 4 to 6 weeks prior to the screener, 24 hours before the screener, and then immediately after the screener, we'll look at a ton of factors, and we'll use advanced mathematics and statistical approaches to do feature selection, dimension reduction, cut it down to a smaller number of factors that are most predictive. And what do those profiles represent? Again, some of these measures are clinical, some of these measures are basic, but the benefits should translate to humans directly, prospectively. And so this is where we get to the basic scientific component. And we're doing this in humans, but clearly the military, and especially right now, there are a lot of elements that are very interested in enhancing resilience, right? How do we improve cognitive performance? How do we improve physical performance? Well, one way to do it is non-invasive brain stimulation. And so if we rewind to 2016, Peter Strick, University of Pittsburgh, discovered cortical projections to the adrenal medulla. They are somatotopic in nature. The vast majority of the projections originate in the M1 trunk representation, which is where the adrenal glands would be. That was very interesting. And this is really the significance of this. It can't be overstated because this is in Cebus monkeys. Surely it exists in humans. And the point is that this explains psychosomatics, right? How our thoughts, behaviors, and beliefs can influence our health and our physiological functions, right? Knowing that the cortex can directly control the output of the medulla. And there are a number of interesting corroborative studies. This is from roughly 20 years ago, some in humans, some in rats. Never replicated, rarely understood, but put out there, right? And the bottom line for this is that these investigators weren't looking for these targets. They weren't looking for these responses. Some of these responses reflect the off-target effects of non-focal stimulation. They were on the pre-supplementary motor area, or they thought they weren't, and they were. They were trying to stimulate one area of the motor cortex, but they were probably getting the trunk representation. Lo and behold, they find that they can modulate corticosterone output, they can modulate time to fatigue in rats, and in humans they can alter the output of the pituitary gland. So some of our initial work, and let's see if this works. So some of our initial work, and let's see if we can, okay, this used to be a video. Well, that's okay. So what this video is demonstrating is a theta burst stimulation transcranial magnetic stimulation protocol, okay? We were targeting the leg region of the motor cortex, and it just so happens that that's right next to the trunk region, okay? It's about 1 cubic centimeter of focality, but what we detected was an increase in a physiological index of SNS output through skin conductance responses, and a reduction in fatigue during physical performance tasks. And again, at the time, this was before these new findings, that was curious to us. Why would we expect to see this? It doesn't really make any sense. Okay, that's interesting. Let's follow up on it. Then the PNS paper comes out, and it started to make sense. And so what we're going to do is establish a tolerability, safety, and optimal protocols for non-invasive brain stimulation targeted at the trunk representation of M1. We'll look at its efficacy, we'll look at a bunch of outcome measurements, and we'll do it in relation to physical and psychological stressors. We'll also explore the possibility of accumulating effects. And this has come up again and again, right? There's a lot of groundwork to do here. So at a high level, we'll have 2 cohorts, right? We're going to talk about localization, targeting, the protocols, the dose-response relationships, its safety and tolerability. We'll look at neuroendocrine factors specific to the hypothalamus, specific to the anterior pituitary, the posterior pituitary, the adrenal medulla, and the adrenal cortex, positive, negative controls. It's basic science, but this was a translational piece that the military was especially interested in. We'll look at physiological measures of autonomic nervous system activity, things like pupillary dilation, heart rate variability, heart rate, respiration rates, skin conductance levels. We'll look at physical performance, we'll look at cognitive function, we'll look at stress tolerance. And of course, we're also very interested, this makes me think of mechanical loading, whether or not the effects of stimulation are present in a resting state or if they require stress, right? Stress combined with stress. That'll be another question. And whether there's any specificity. Are the responses larger in the presence of cognitive stressors, physical stressors, or does it depend on the population, right? Subtypes. So, the first thing that we need to figure out is whether we'll have everyone do an MR scan and an FMR scan with imagery, whether we can use an atlas, and I mean this goes all the way back to Penfield and Woolsey, right, and before that. But whether we can use an atlas and prior knowledge about the structure of the brain to target the M1 trunk representation, whether it's better targeted with changes in blood oxygenation levels in the brain, or if we can do it functionally based on motor evoked potential responses to stimulation in vivo with frameless stereotactic neuronavigation. Once we determine the optimum really area to look to stimulate, exactly what we want to stimulate, the question is, do we produce the same responses when we target M1? Do we produce different responses when we target the pre-supplementary motor area? Do we produce different responses with a subthreshold patterned stimulation? This is just an example of theta burst stimulation. It's triplets at 50 hertz. They're repeated at theta frequency. There's 10 of them separated by 8 seconds of rest. It's subthreshold. There are about 4 synapses between the cortex and the adrenal medulla. And so the question is, can changing the activity of cortex alone modulate the output of the adrenal medulla, or do you need suprathreshold stimulation, right, to pass through all of these synapses and actually activate those neurons directly? And this would be, as an example, a 20 hertz excitatory protocol. Another question, we know that different populations of neurons display different sensitivities based on the direction of currents. So, do we consistently or individually produce different responses depending on the type of coil orientation we use? And of course, what are the responses in respect to dosages? We know that the brain is interesting, right? You do anything and it responds, and sometimes it responds in a paradoxical way. So it isn't entirely evident that increasing the dosage or intensity will lead to a linear increase in the output of the adrenal medulla. It may have the opposite effect, and in some individuals that may be beneficial. So we need to take a look at that. And then, this is just 2 examples. Exercise, this is a well-established intense exercise, a well-established stimulus for neuroendocrine output, whether that modulates the effects of stimulation, whether we can use increased catecholamine output, increased neuropeptide Y output, brain-derived neurotrophic factor output, to increase time to exhaustion, to increase maximal force production, these are in healthy participants, or to increase the time until participants basically withdraw from a cold pressor test. They just have their hand in an ice cold bucket of water. It's unpleasant, it's painful, it's non-voluntary in respect to exercise, and so we measure the time until participants withdraw, right? Does it take longer? That's especially relevant to the Navy because if you're a SEAL, you spend a lot of time really wet, really cold, and really uncomfortable. If we determine that this is safe, tolerable, and it works, the next question is, are there accumulating effects, or are they attenuated over time? That's a possibility as well. Maybe there's a response to the first session, and after that, they all go back down to nothing, and we don't see anything anymore. Maybe neuroplasticity works against us, or the results are amplified. That's a possibility. And if that's the case, we need to know about it. So we will subject participants, assuming that this works, to repeated sessions of non-invasive brain stimulation to determine what the therapeutic effects look like over a chronic period. And when I say chronic, I'm talking about 6 sessions, not 6 months at 3 days a week, right? So this is preliminary work. One of the things that got the Army on board was the technological innovation piece, right? There are concerns about the focality, depth, power trade-off, transcranial magnetic stimulation. This is nothing new. So we're partnering with the inventor of the transcranial magnetic stimulation coil to develop a coil that penetrates more deeply but retains its focality, and it's actively cooled, which allows it to deliver high amounts of energy repetitively over a long period of time without overheating. You're always fighting physics with these kinds of things, and that still limits transcranial magnetic stimulation and direct current stimulation to this day. And of course, there will be room for questions about whether we can translate this to an electrical paradigm, because heck, you've got people making them in their garages right now, and there's going to be a lot of interesting headlines over the next 10 years. But you know, they're low cost, they're mobile, they're portable. So yeah, there's appeal, especially in relation to like a series of 3 capacitors with thousands of volts and huge cables and all of the stuff that comes along with the transcranial magnetic stimulator. So that's one of the things that we're thinking about down the line. And again, one of the things that they always want to know is, well, what can we do to automate this? We may not have you or an expert on site somewhere in Afghanistan. How do we automate the analysis to know that the responses are what we expect? Is it mobile? What is the ease and the repeatability of administration? How do we get the cost down? How do we minimize patient input? And how do we make administration as fast as possible? And in many cases, we're talking about building pieces of equipment that allow us to do that better, allow us to do it better. So that's, you know, again, that is part of the translational piece here. I mean, we can use this for basic scientific questions in humans, but we need to be able to kind of translate it to a model where it can be used in a field setting. And I mean, we spend a lot of time going back and forth with program managers about this thing. I mean, everyone's cognizant of it, right? You have to be able to show the impact and the significance for whatever it is you're doing, however promising. So one of the collaborations that came forth from this was work that will be done at the Aberdeen Proving Grounds. And this is, oh good, I'm doing all right. This is specific to special forces. There are virtual marksmanship platforms that allow you to look at team decision-making, rapid decision-making. There are wireless FNIRs and EEG systems that will allow us to look at specific brain processes, and obviously we can measure neuroendocrine responses. But assuming this works in a laboratory environment, we'll be able to translate it to the environment that the military cares most about, which is a raid. You know, a team going on a raid, making the right decisions as quickly as possible, distinguishing friend from foe, and everyone coming out safely. That's what we need to know. So I appreciate your time, and that's it.